Remember, poliovirus is in the enterovirus genus in the picornavirus family. And now we're going to talk about the other viruses in the enterovirus genus. And we say other, but really polio is almost eradicated, whereas these other viruses are absolutely everywhere. You've all had them, and you'll all see them many times, especially among kids. So what are they? Let's start with the name game. They include echoviruses, coxsackieviruses, and enteroviruses. Yeah, the naming here is a little annoying. Enterovirus is the genus, and there are species in that genus called enterovirus. And there are also many, many subtypes of each of these, echoviruses and coxsackieviruses. And you don't need to know all the subtypes, but you do need to know that coxsackieviruses are usually divided into two groups, Coxsackie A and Coxsackie B. And you need to know that because when you test for Coxsackie, you usually test for A and B separately. So let's talk about what the enteroviruses can do. So like polio, infection always starts in the GI tract with or without symptoms and then spreads. So where can it go? First of all, the skin. There it can cause hand, foot, and mouth disease. And this is usually caused by Coxsackie A, and the symptoms are easy to remember based on the name. It gives you a rash on your hands, feet, and your mouth. And the rash usually consists of small tender lesions, either vesicular or maculopapular. So by itself, this is no big deal. You can also get other rashes, lots of different rashes from various enteroviruses. The next spot that enteroviruses can go are the eyes, where they can cause acute hemorrhagic conjunctivitis. And in this case, the virus infects the conjunctivae, which are the mucous membranes lining the eye. And it can cause bleeding underneath those, which causes pain and makes your eyes look blood red. And usually this heals on its own and doesn't cause vision loss or permanent damage. Now, getting more serious, enteroviruses can also go to the heart, where they can infect muscle fibers, which we call myocarditis. And this is very important because it can cause heart failure in someone who's young or who has no ischemic heart disease, and Coxsackie B is usually the most common culprit. Finally, enteroviruses can also go to the central nervous system, where they can cause several diseases. So most importantly, they can go to the meninges and cause meningitis. And in fact, enteroviruses are the most common cause of aseptic meningitis, meaning non-bacterial meningitis. Now, more rarely, these viruses can infect the brain itself and cause encephalitis. And last of all, remember that polio is an enterovirus and causes paralysis. And the reason it does so is that it likes to infect the anterior horn cells of the spinal cord. Well, it turns out that other enteroviruses like to do that too. And so they cause what we call acute flaccid paralysis. Enterovirus D68 is often implicated in this. So the obvious question is, why do we seem not to care as much about this as polio? And the reason is that these people usually recover, unlike those with polio, and that's why it's called an acute flaccid paralysis. It doesn't cause permanent paralysis. Now, the last thing that enteroviruses can do that you should know about is they can infect newborns, in which case they can cause an especially severe form of disease. And the reason for that is that newborns can't mount a normal immune response. They can't make their own antibodies. Usually they're protected by maternal antibodies. But if a pregnant woman gets an enterovirus infection just before delivering, she could potentially infect the baby but not have time to make and provide protective antibodies against that virus, because those take a while to make. And babies in this situation are at really high risk. They can get disseminated infections of the heart, brain, liver, and more. So those are the most important presentations of enteroviruses.
Welcome back to Med with Med Simple. Support me by donating me on Patreon. Subscribe to my channel for more videos. Thank you so much for 400 subscribers. This video is about coxsackieviruses. Coxsackieviruses belongs to a family known as picornaviruses. There are two main pathogenic genus in picornaviruses, namely enteroviruses and rhinoviruses. Coxsackieviruses comes under enteroviruses. Let's see the general description of picornaviruses. They are RNA viruses, spherical in shape, they are non-enveloped, and they have viral proteins in their capsid. How are they transmitted? The most common route of transmission is fecal-oral route. When you drink contaminated water containing the viral particles, you can get the infection. The other common route of transmission is by respiratory aerosols, which spread in the respiratory droplets from infected patients. The incubation period of coxsackie-viral infections is about 2 to 9 days. There are two main groups of coxsackieviruses, namely Group A coxsackieviruses and Group B coxsackieviruses. Now, how do they classify the coxsackieviruses into two separate groups? Initially, they did some experiment in the suckling mice. What they did is they injected the virus particles intracerebrally into the mice and they observed the changes. There were two common changes, namely spastic paralysis and flaccid paralysis. Based on that, they classified coxsackieviruses into Group A coxsackieviruses and Group B coxsackieviruses. Before continuing to watch this video, please leave a like and subscribe to my channel for more videos. Now let's talk about Group A coxsackieviruses first. When injected intracerebrally into the mice, they produce flaccid paralysis in the mice. In humans, few serotypes of Group A coxsackieviruses can cause aseptic meningitis. They can also cause herpangina, in which there will be vesicle-like blisters in the oral cavity and pharynx. A very important thing to know about Group A coxsackieviruses is that they can cause a disease known as hand-foot-mouth disease, in which you get vesicle-like blisters over the palms, feet, and in the oral cavity. They can also cause acute hemorrhagic conjunctivitis, in which there will be congestion and reddish discoloration of the conjunctiva. Group B coxsackieviruses can infect any age group. They most commonly infect the children particularly. When injected intracerebrally into the mice, Group B coxsackieviruses will cause spastic paralysis. All serotypes of Group B coxsackieviruses can cause aseptic meningitis in humans. They can also cause pleurodynia, which is chest pain, myocarditis, which is inflammation of the myocardium, hepatitis, which is inflammation of the liver, pancreatitis, which is inflammation of the pancreas, which can be so severe to an extent that it can cause diabetes in children, and they can also cause pneumonia. You can see the fact that Group B coxsackieviruses can almost involve any system in the body. Lab diagnosis. How do you diagnose coxsackie-viral infections? First of all, we need to collect the specimen. The appropriate specimen has to be collected for the appropriate infection. For example, in herpangina, where there will be vesicle-like blisters or the oral cavity and pharynx, specimen has to be collected from throat swabs and from the lesions. So according to the site of infection, the appropriate specimen has to be collected. The virus can be isolated by various methods. One of the methods which can be used is intracerebral inoculation in mice. As I told you earlier, when you inject the virus into the brain, that is intracerebrally into the mice, what can happen is that they can produce either flaccid paralysis or spastic paralysis. If the virus produces flaccid paralysis, they are Group A coxsackieviruses. If they produce spastic paralysis, they are Group B coxsackieviruses. Another test which is highly sensitive and highly specific is polymerase chain reaction, which is abbreviated as PCR. This is one of the very commonly trusted tests, because the results are highly sensitive and highly specific. Serological tests can also be used, in which we actually detect the antibodies against the viral particles in the patient's serum. We can detect specific rise in antibody titers such as IgM and IgG. IgM titer rise denotes a current infection. IgG titer rise denotes a past infection. Help me to make more videos by donating me on Patreon. The link is given in the description of this video. Please check it out. We came to the end of this video. Please leave your valuable suggestions in the comments below, and I'll see you in my next video. Subscribe to my channel. Thank you.
Bovine spongiform encephalopathy, or BSE, more commonly known as mad cow disease, is a progressive neurological disorder of cattle that results from infection by an unusual transmissible agent called a prion, composed of the cellular protein PRP. Scientific evidence has led to the widely accepted theory which holds that PRP, found in highest concentrations in the plasma membrane of neurons, is normally in a stable shape that does not cause disease. This protein can be flipped, however, into an abnormal shape that does cause disease. The process occurs exponentially, meaning a single infectious PRP can convert many more PRP to the infectious form. Prions can be transmitted through consumption of infected meat and by inoculation directly into the brain or into muscle tissue. The subsequent exponential amplification of the infectious prion would then result in disease. Humans cannot contract BSE, but in rare cases, they can contract a human form called variant Creutzfeldt-Jakob disease, or VCJD. This can happen through the consumption of nerve tissue, such as the brain and spinal cord, of cattle that were infected with BSE. We do not yet know why the abnormal structure of an infectious prion would result in neurodegeneration, but we do know that abnormal PRP accumulates in brain tissue. One part of the prion protein can cause neuroapoptosis or programmed cell death through the entanglement of neurons and interference of synapse function. As the neuron cells are damaged and lost, tiny vacancies form in the brain, giving the brain a sponge-like appearance. Eventually, enough neuron cells are lost to result in the death of the organism. Research indicates that the first probable infections of BSE in cattle occurred during the 1970s, with 2 cases of BSE being identified in 1986 in the United Kingdom. The possible origins of BSE are found in the cattle consumption of meat and bone feed that contained BSE-infected products, from a spontaneously occurring case of BSE or scrapie-infected sheep products. The disease was amplified and spread throughout the United Kingdom cattle industry by feeding prion-infected feed to young calves, thus allowing the disease to survive and expand. Symptoms of mad cow disease in cattle include nervousness or aggression, coordination problems, acute muscular twitching, difficulty standing up or walking, and death. The BSE epizootic in the United Kingdom peaked in January 1993 at almost 1,000 new cases per week. Since then, the annual number of BSE cases have dropped sharply. Cumulatively, through the end of 2015, more than 184,500 cases of BSE have been confirmed in the United Kingdom alone in more than 35,000 herds of cattle. Outside of the United Kingdom, BSE has been most prevalent in nearby European nations, such as France and Portugal, during the outbreak. Scientific evidence for association between the human prion disease, VCJD, was first reported from the United Kingdom in 1996. The interval between the most likely period of extended exposure of the human population to the potentially BSE-contaminated food from 1984 to 1986 and the onset of initial VCJD cases from 1994 to 1996 is consistent with known incubation periods of the disease. The disease can affect all age groups and is difficult to diagnose until advanced stages. Over time, VCJD destroys the brain and spinal cord. In the early stages of VCJD, people have symptoms related to the nervous system, such as depression and loss of coordination. Later in the disease, dementia develops. VCJD is usually fatal within 13 months of the onset of symptoms. Since prions do not contain nucleic acid, they are resistant to most pathogenic treatments that target genetic material. In addition, infectious PRP is a malicious form of a naturally occurring protein in the organism, so it does not trigger a host immune response, making them virtually invincible. Thus, treatment for VCJD remains supportive only. No specific therapy has been shown to stop the progression of this disease. Since 1989, to prevent BSE from entering the country, the United States government has prohibited the importation of certain types of live animals from countries where mad cow disease is known to exist. Prohibiting high-risk animals from entering the food supply and the removal of central nervous system tissue from the food supply also helps minimize the risk of BSE and VCJD to consumers.
Di urutan keempat ada rabies. Rabies merupakan virus mematikan yang dapat ditularkan dari air liur hewan kepada manusia. Pada tahun 2009, 93 anak di Angola meninggal akibat virus ini. Rabies dapat ditularkan melalui sejumlah hewan seperti kucing, kelelawar, anjing, kera, dan luwak. Virus ini menyerang sistem saraf dan otak manusia. Jika sudah mengalami gejala-gejala virus ini, risiko kematiannya bisa mencapai angka nyaris 100%.